confirm it's 12:06. It's 12:06. Okay. And trip explanation. We're doing Friday. Not today. Okay. Oh my God. Hi, everyone. We're going to start in just a minute. I'm going to ask from now on that you actually have your video on. So if you need to walk away for a second and do something so you can come back and be presentable, go ahead and do it. But I'd like everyone to have their video on during class. So, Rob, what is it? The question that you wanted to ask me?
The question, I think it's for your bio class and not for us. It asks about gene molecules in your G1.
No, it says here, can someone ask about the attendance question?
Oh yeah, so I don't know if it's the right question for your forensics class. It says what are two genes that would be transcribed through the cell cycle? Two that would transcribe specifically in G1.
I sent that to you guys by mistake.
Yeah.
I did. And did I send them the blood type question? Oh my God. Did I send any of you the blood type question?
No.
Oh,
No.
okay. Sorry.
Hey, I have the blood type questions.
Yeah. Amy, I need to see you, actually.
One sec.
But you. Guys, I need everyone has to have their video off. If you need to step away quickly, put on something. Sorry?
Dr. Hornstein, some people said that they don't have a camera in the chat.
You don't have a camera anymore. Okay. Okay. Well, I expect if you have a camera, you'll put it up. Thanks. Okay. So actually, the question I meant to ask you was, what are the 4 blood types and which is the most prevalent blood type? So that was actually the question I meant to ask you. The other one was for my other classes. So, so anyone wanna anyone want to give that a shot? A, B, and O. Good. Good. Absolutely. And O is the most. Okay. Okay, good. We're going to get to that in a few minutes. So here's the plan. A couple plans. Number one is, I'm going to send you after class today. I'm going to send you a questionnaire. We've been normally that what we do is we go on a trip to the DNA Learning Center, but obviously we're not doing that in April. So they're going to do a virtual trip for us. And it's DNA fingerprinting. If you guys remember from freshman year, something called short tandem repeats, which is how we, when they do DNA fingerprinting at a crime scene, they're looking for 13 different short tandem repeats. And we're going to actually, what we normally do is we extract our own DNA and look for one of those. But they're going to do that for us. It's an hour exercise. So what I need to know is, and I'm going to send the questionnaire, we can either do it 2 and 3, 3 and 4, 6 and 7, or 7 and 8. And it'll be 2 sessions. So it means that if you normally see, so the bulk of it would be either 3 or 7, and then there'd be a little of 2 or 4 tagged on, or a little bit of 6 or 8 tagged on. So I'm just going to send you a questionnaire to see what is the most number of people which is the most popular of those choices, okay? So if you would send that back to me by the end of the day, I'd appreciate it. Any questions on that? Okay, I'm also going to say, as I said beginning, if you have a camera, I'd like to see your faces rather than your names or a photograph. So again, if you have to make yourself presentable and walk away and come back, I get it, but do that, okay? And the last thing is, I thought, let's get back into some work today, and you can be working on, and maybe on Friday I'll break you out into groups to work on your project. But I'd like to do I'd like to try to finish the work on blood by the end of the week so we can move on, okay? Any questions about the assignment that have come up? Anything that's come up about the assignment? Okay. Okay, so then let's just get right to it. And you're going to need to take notes for this. Okay, so, oops. So give me a second. Okay. So, can you guys see that okay?
Yeah.
Okay, good. So again, the attendance thing is, remember every day, because not everyone can make this period session, they want us to keep track of you and I want to keep track of you. So just answer the question. And for those of you who came in late, I gave you my freshman question, and I gave my freshman your question. I think, what was the question I gave you? I forgot already. What was the question I gave you again? Does anyone remember?
The question is, which two gene is always transcribed in the cell cycle?
Oh, I see. Okay, good. Okay, so yeah, so we're talking about the cell cycle right now. So we're entering transcription. Okay, good. So here we go. So what I want you to do now is I'm going to give you 3 minutes, and I want you to answer these 3 questions. The first thing I want you to do is I want you to write down as many components of blood as you remember. We're going to be talking about blood not as from a spatter point of view, but from a component point of view. There's other ways that blood composition can help in a crime. And then tomorrow we'll be talking about semen and saliva. Okay, so write down at least 6 components of blood. Which of these components could be used in a forensic case, and which would be considered, of those components, which would be considered class evidence and which would be considered individual evidence. So take 3 minutes actually write it down for yourself on your notes first, and then we'll share out on here. But write it down for yourself. Go ahead. Just 2 minutes. 2 minutes is enough time for that. And I would also appreciate you keep your mic muted when you're not talking. I just hear chewing or something, and I get it, but if you would do that, I'd appreciate it. Thanks. Thanks, Denise, for writing that out for me. Okay, just 10 more seconds. Okay, let's see how it works verbally. We're going to experiment. So we've been doing the typing thing. Now I want you, let's just do a little bit of verbal stuff. We're going to go back and forth and experiment. So give me 6, just tell me a component of blood. Go ahead. Anybody?
Plasma.
Water.
Plasma. Good. What is plasma? What is plasma? What's the definition of plasma? Anybody? Okay, it's a non-cellular component of blood. Everything except the cells. Okay, what else besides plasma? Go ahead, someone else.
Platelets.
Platelets, good. What do platelets do?
They clot blood.
Good, they cause blood. Okay, give me another component.
Red
Red
blood
blood
cells.
cells.
Okay, I heard red blood cells or erythrocytes. And what do? What's their main function?
Carry oxygen.
Good. Yep. And I heard white blood cells. And what's the scientific name for that? That's leukocytes. And what is the major function of leukocytes?
Immune system.
Right, they're all part of the immune system. Good. Okay, what other components? Give me something. That's just great. Give me some other things.
Antibodies?
Antibodies, absolutely. Good. What else?
Hemoglobin.
Okay, what do you, well, hemoglobin is located inside what?
The red blood cells.
The red blood cells.
Red
Good.
blood cells.
What else?
Iron.
Iron. That's also located in the red blood cells primarily, though there is some, but that does bring up ions. There's ions floating around your blood. There's iron, there's sodium, there's potassium. That's absolutely. What else? What are the things that are involved in, what do you guys have a lot of in, well, you guys are having less and less in your blood as you go through adolescence, as you're finishing up adolescence. What's in there?
Plates.
Hormones?
Hormones, right? Hormones are there. Waste product is there. What else is there? What else is there? All the nutrients, right? The nutrients. So there'd be what? There'd be glucose, there'd be amino acids floating around like that. Okay, good. And one more. What would be an unusual thing that would be in there that might be really
Drugs.
useful for a forensic scientist?
Drugs.
Drugs, right. So any toxins that are there. Good. Okay, good. So which of these components, so toxins obviously really useful for a forensic scientist. Give me an example of when a toxin would be a really nice piece of evidence for blood evidence in forensics.
Maybe like a rape case, like a drug rape case.
Sure, if there's a the date rape drug, it would be absolutely good. Okay, what else of these components would, sorry, go ahead.
Determining cause of death.
Determining cause of death. Good, good. Right, so if you have arsenic in the blood or something like that, right? And you guys did a case like that at the beginning of the semester. Anything else that could be used by forensic scientists?
If the patient has like a disease.
To see if they have a disease, some sort of bloodborne disease. Okay, so say more about what that might look like in a forensic case. Give me an example.
I guess just to establish like the the victim's like background for again like cause of death. It might have been.
Okay, good. Right. Okay, so
that.
someone might have died from natural causes because they have some infection or some disease, a genetic disease maybe, and you could maybe use that to rule out that there's any that they've been murdered, for example, and that in fact they died of natural causes. Yeah, good. One more, the big one.
White blood cells.
Thank you. White blood cells.
They contain the DNA.
That's right, they contain the DNA. The other two do not contain the DNA. Good. So of all the stuff you said so far, what would be considered class evidence? What would be considered individual evidence? So class evidence means it brings you down to a class of people. And individual evidence, it makes it only one person has this. So what would be class evidence? Give me one example of class versus individual that we just talked about for number 2.
Blood type would be class.
Good, blood type would be class. And what would be individual?
DNA.
Blood cells.
DNA.
DNA.
White blood cell.
Yeah, the DNA, absolutely. Good. Okay. Okay. So this is called serology, specifically forensic serology, right? The study of serum. And there is the unit in the crime lab is called forensic serology, and it handles blood, semen, and saliva. That's all in one unit, right? So we have blood, semen, and saliva. So what is blood? It's a complex mixture of cells, enzymes, proteins, inorganic substances. We didn't talk about all those enzymes and proteins that are, well, we did, we mentioned antibodies, but there's other proteins that are floating around the blood. It is the transport system of the body, bringing nutrients to the cells and removing waste from the cells, right? It's a major component. Again, someone, it sounds like they're typing or eating. I can't tell what. I'm sorry. If you could mute it, I'd really appreciate it. Thank you. So yeah, bringing new nutrients to cells and removing waste from cells. And we talk about the serum. The serum is the fluid portion. It's similar to the plasma. It's the fluid portion of the cell after it clots. So you take, this is blood before it clots, this is blood after it clots, and what you have left is the serum. It's roughly 55% of the blood content. It contains the proteins, the nutrients, the electrolytes, etc., but no clotting factors. The clotting factors are removed when the blood clots. And then we have plasma. Plasma is the same thing as red blood, as serum, except it also contains the clotting factors. So if you want plasma, you don't cause it to coagulate. So, but what you do instead is you put it in a centrifuge, like here, and you spin it down. So this is before it's been spun down in the centrifuge. This is after. These are the cells. Specifically, these are the red blood cells, the white blood cells, and the platelets, and then this is the plasma here. And that's what it looks like here. So that's the difference between plasma and serum. Generally speaking, you can add anticoagulant. If you're working with plasma instead of serum, then you generally need to work use an anticoagulant. And again, this is before and after picture of that. Okay, what are the types of cells? Well, we have the erythrocytes, or the red blood cells. They have no nucleus. They contain hemoglobin, which is important. We're going to see in a second. Is the group chat in the way for you as it is for me? It must be. If it is for me, it must be for you. If I can move that there. Oh, no, it's not what I meant to do. Okay. So it's hemoglobin is transporting oxygen from the lungs, sorry, to the cells, and the hemoglobin gives the blood its red color, right? And that's just a picture of hemoglobin. And hemoglobin is made up of.
Dr. Ortiz, can we slow down a little bit?
Sure, sorry. Oh, while you're taking notes, someone, what's the color of a deoxygenated, oxygenated blood is bright red. Does anyone remember what the color of
Blue.
deoxygenated blood is?
Blue.
No, no. That's the color of actually the vein. That's the color. That's the side of the veins. That's not the blood itself.
Black.
Like no,
Brown?
maroon. You guys know what maroon looks like? Like a dark red. It's like a dark red. Yeah. Anyway, so yeah. Okay, and that's a picture of hemoglobin, and we mentioned that it has iron in it. It has 4 proteins. It has heme, and it has iron, and there's 4 of them, and there's an oxygen binding site in each of these 4 places. Then we have white blood cells, or leukocytes. Cancer of the white blood cells is leukemia. These have the DNA, and that's what's important for us. And they're a member of the immune system. And there are 5 important types that battle pathogens, mostly bacteria and viruses. Also, they're involved in allergies and in the inflammatory response. And for us, I'm sorry, is that too fast? For us, there are really 2 major types. For us, what's really, sorry, what I meant to say is for us, what's interesting to us is the DNA. But there are 5 different types. There are the eosinophils, the neutrophils, the base. You do not have to write this down. The basophils, the lymphocytes, and the monocytes. You have granulocytes, which tend to secrete things that destroy the neighboring cell by secretions that eat away at a cell. And then you have macrophages, which tend to engulf cells. Those are the 2 major types of killing that take place. So there are others. What does a forensic scientist learn from blood? Well, we're going to talk about a few things. First of all, the first question a forensic scientist has to ask if they see something that looks like blood, and this is so pertinent, as you know, to what you're reading about now, is you have to first make sure that it's blood, okay? And then the next thing to make sure is that it's human blood, because we're not so interested in it if it's not human blood. And lastly, can we in some way tie it back to an individual? And if we can't tie it back to a specific person, can we at least tie it to a group of people? Okay, so let's take a look at this. So the first big question we have to ask is it blood? And there's a bunch of presumptive tests for that. And all of them are color tests. And when they go to a crime scene, the forensic scientist will be carrying at least one of these chemicals with them: phenolphthalein, leucomalachite green, and luminol. And each one is a test for the presence of hemoglobin, and we'll see how that works in a second. The Kastle-Meyer test, the phenolphthalein, it turns red in the presence of hemoglobin, like this. Leucomalachite green turns green in the presence of hemoglobin, and luminol turns blue in the presence. Okay? So what is the science behind this? What's the mechanism for this? So what you have here, you have an uncolored reagent, right? And you add hydrogen peroxide as well. So we have one of the three reagents that we just mentioned, plus hydrogen peroxide. Hemoglobin has enzymatic activity, and it's capable of transferring the oxygen, one of the oxygens, onto one of these reagents. And when it transfers the it doesn't itself do it, sorry, it catalyzes this, but the transfer of oxygen here causes that to change color to one of those three colors, and then you produce water. So hemoglobin is a catalyst to the oxidation reaction of hydrogen peroxide. Hydrogen peroxide donates, hydrogen peroxide is being reduced. Hydrogen peroxide donates one of its oxygens to the reagents, and it turns color. And just to refresh your memory, right, a catalyst is a substance that speeds up a reaction without, in fact, being used up in it. So hemoglobin is not reacting here, it's just catalyzing this reaction. Some things you have to think about as a forensic scientist: these tests will destroy the blood, so you won't be able to use it for further testing. So therefore, you have to be careful. You can't use up all the blood, because these are presumptive tests. They're extremely sensitive. They can recognize blood in a 10,000-fold dilution. And we talked about, like, with the footprints, you could see like 200 footprints out in blood. When you have luminol at the crime scene, you can spray and find that out. But also, because they're presumptive tests, they have false positives. So vegetables, horseradish, potatoes, and some metals could all give a false positive. So therefore, when you get back to the lab, you have to save some of that blood, that presumptive blood, and make sure that in fact is blood, and you do what's called a crystal test. Forensic scientists do one of two crystal tests to prove that something is blood, and you don't need to remember which is which, but one is called the Takayama test and one is called the Teichmann test. And what it does is, in both cases, they form these very specific hemoglobin, when in the presence of certain reagents, will form very specific, these very long, thin crystals. And so if you get these long, thin crystals, you know for a fact then you have blood. Okay, the next question is it human blood? And what you can do is, you want to do a species test for blood, and to do that, you do what's called the precipitin test. Okay, and these are different blood. So this is bird blood, fish blood, and this happens to be dog blood. Okay, so the precipitin test, sorry, I'll leave that up. So the precipitant, okay. The precipitant tests is using an antibody. And let's just refresh our memory about what an antibody is. An antibody is a protein that binds to a specific antigen. This is what a picture of an antibody looks like. This is a cartoon of an antibody. This is the constant region. This is the variable region, the two arms. So an antibody will bind these two sides of the variable regions. This is where they bind what's called antigen. The antigen is the molecule, the specific molecule that an antibody will bind to. And these two are identical, so they bind the same antigen. And every antibody binds a different antigen. That's what makes it so powerful in our body and as a tool for scientists. So every single antibody binds a different one. They're found in the serum of the blood. We have thousands or hundreds of thousands of different antibodies in our blood to recognize any foreign substance that happens to be there. And this is what happens when antibody recognizes an antigen. So this is called agglutination. These purple guys are the antibodies, and the round circles are cells with antigens on the surface of them in this case. Okay? And remember, each of the purple guys binds the same antigen, and there's multiple antigens on the same cell. It doesn't have to be a circle. A cell, this could be a molecule as well. It doesn't have to be a cell. And so what happens is you get clumping. Whenever you have antigen and you add antibody that's specific for that antigen, you get clumping. And these can be millions of molecules long. Okay? And we call this clumping of an antibody-antigen complex agglutination. Agglutination is clumping. Now, when this happens in the body, when this happens in the body, then macrophages recognize this complex and they come and they just devour it. They'll eat the entire complex. And that's how the antigen is destroyed. But when we're using it scientifically, we get agglutination and we get clumping. So before we look at an example of that, I want to ask you, what is it about the structure of the antibody that allows for clumping to take place? What is it about the structure of an antibody that allows clumping to take place?
The proteins on the outside.
That's right. What about, okay, good. So that there are proteins on the outside of the cells. Okay, and what else? What about it? What about it? Can you say more, Gordon?
Well, the antibodies or specific antibodies are made by the like blood cells that can attach to the protein receptors and then like flag down the other parts of the immune system.
Right. Yes, that's absolutely nothing that you said there is untrue. I was asking about the clumping, and I'll just tell and why it clumps so much. And the reason you get the huge complexes, which is because you have multiple antigens on the surface of the cell here, but the antibody has two arms. If the antibody had only one arm, if you can reimagine this picture, you'd get a bunch of cells coated with individual antibodies, but the cells would not be themselves attached, right? But what makes this all one big, huge complex is the fact that every antibody has two arms, so it can bind to two different cells. And in doing so, you get this huge amount of clumping. Okay, and sorry, and this is, so here we have agglutination on the bottom here. We have 5 things, and 3 of these are agglutinated and 2 are not. So what we're seeing here is, here's agglutination or not agglutination. What do you think, guys? Agglutinated or not agglutinated? Compare these 2. Which is agglutinated and which is not agglutinated? Left or, let's just talk left, right. The one on the left or the right is agglutinated. Is this agglutinated or is this agglutinated? Hold on. Hold on 1 second. I'm trying to. Give me a second. sorry. I've lost the chat. I wanted to get the chat back. Everyone's so shy. You're much less shy. Okay, we'll just keep going. This is agglutinated, and this is not agglutinated. And what you can see here, the clumping is here, right? It's coming out of solution, and then you have all the clear plasma over here. Whereas here, it's more diffuse, right? The red blood cells are more diffuse here. So here you're getting clumping, and here you're not getting clumping. Here's a more dramatic version of it. This is unagglutinated, and this is agglutinated. This is blood sample, right? You're getting agglutinating of the blood and non-agglutinating of the blood, just like in that picture, just like in that, right? Okay. The making of antibodies, it's really cool. You can make antibodies against anything you want. So for scientific or forensic purposes, you can make antibodies against anything. And all you do is you basically just inject it into an animal. You inject a particular molecule or substance that you want an antibody against into an animal. They use rabbits, they use goats, and then you just drain the blood and you collect the plasma. And what you're going to have is a whole set of antibodies that bind to that particular antigen. And then you can purify them, and then you can make monoclonal antibodies, which are only against one antigen. But the gist is you put it into an animal. Okay, now coming back to our original test, we were talking about how do you define whether it's human blood or animal blood, and that uses something called the precipitin test. And this is a precipitin test because you get precipitate. Now, what does that mean? To precipitate, if you have a solution that forms a precipitate, it means that some solid comes out of solution and forms at the bottom of the. If you have a glass beaker with a clear solution, and then you induce precipitation, then what you're going to get is a solid substance that forms at the bottom of the beaker. So this is a form of forming a solid, and the solid is really this agglutinated substance with the antibodies and the antigen. And so what do they do for this? There's different ways that you can perform this test. If you take a test tube and you layer in on the bottom antibodies, and then you layer in on the top the substance that you're trying to test. You layer in on the top the substance that you're trying to test. What's going to happen is in the middle, you're going to get agglutination, because what's going to happen is the antibody is going to bind the antigen, and in the middle, you're going to get a precipitate where this clumping takes place. And you're going to get a little band that looks something like that. Okay? And that's how you know that you have it. So this is what a positive reaction looks like, just so now let's look at an actual example. So here's what they will actually do. They'll take a gel and they'll poke a series of holes in it. A series of wells. There'll be a well in the center, and in the well in the center, they'll take the presumptive blood from the crime scene and put it there. And then what they'll do is they'll take a bunch of different animal blood. They'll take, let's say, here is human blood, antibody against human blood, and this is antibody, against dog blood, and this is antibody against cat blood, and this is fish, and a bird, and deer. And then they put it in there, and then they let it sit. And what happens is the antibody will diffuse in all directions out of here, so it'll diffuse there, and there, and there, and there. And the blood, the antigen in the crime scene, will also diffuse out there, and there, and there, and there. And there'll be an interaction where they meet in the middle. So my question to you is, what kind of blood is this? What kind of blood is this? Based on that idea, and this is where I would love the chat room, so I'm going to try to pull it back up. Hold on one second, okay? What happened to the chat room? Okay, hold on. Okay, minimize. Okay, screen share. And where are we? Okay, up here. So, okay, good. So, anyway, now here, you can either type it in or you can say it. What kind of blood do we have here? What animal? No, I'm sorry, let's see. No one? No one? Okay. Okay, so this here is the precipitate. You have to, they're trained in this, right? This is the precipitate. So therefore, this is human blood. So the antibodies diffused here, the human blood or the substances in the blood diffused here, and here you get the precipitate. So this is human blood. So that's basically how that's the species test. And then the last thing you do is to classify the blood. Not the last thing. And there are different blood types, as we know. There are over 250 blood types that exist. The ABO, which is one we're most familiar with, but there's CDE, CE, MN. And we're not interested in those guys because, I'm sorry, all of these represent different types of proteins on the surface of a red blood cell. But we're only interested in the ABO and the RH. And the ABO and the RH are the most important for us because those are the only ones that are that are important immunologically. We tend not to have immunological reactions against these other guys. So we're only interested in this blood typing and this blood typing. Now, I see that we have to go in a minute, so let me just stop for a second. I'm thinking, I put in an assignment. Yeah, I put in an assignment for tomorrow. So I'm just going to stop here for a second. So we're up at blood tests. Class ends, I believe, it started at 12:06. I think it ends in like 1 minute. So oh, Amar, you didn't get the last thing? Oh, you didn't get you didn't get the precipitin test?
No.
Okay, I'll make sure to go over it tomorrow because I think I don't want to keep some of you. So I'll make sure to start with the precipitin test tomorrow. But I just wanted to say, the case you're gonna, it's a 15-minute video and you're gonna answer a few questions about the video. It's a very famous case in Australia from the 80s that was about, it was a serology case. And it was very controversial and we'll talk about it tomorrow. So any questions? But I'm sorry, I went further than I expected to.
Yeah, are we supposed to do anything with the worksheet you posted?
Yeah, that's what I'm talking about. Wait, which worksheet?
The one with the blood types.
No, that's for tomorrow's. Thank you for asking me. That's for tomorrow. I thought I would get further today in the lecture. That's for tomorrow's lecture.
Oh, okay.
The only thing I want you to do for tomorrow for homework is to watch that 15-minute video and answer the questions about it.
Okay.
The Azaria Chamberlain murder case. Okay.
Wait, what about the attendance assignment for today?
What about it?
Are we answering the do now questions?
Yeah, answer the do now question as best as you can.
Okay.
Just it's yeah, just answer it as best as you can. Any other questions about it?
What were the do now questions?
Yes.
Well, the do now questions was how many blood types were there?
There was, what were the different blood types in the AB? I meant the ABO system. I mean, what were the blood? I was referring specifically to that. And then what was the most prevalent? That was the question that was asked. But that was what I was supposed to ask you, but instead, so if you want to go ahead and answer that. You know what? As soon as the class is over, I'll put a message in with that question. You can answer that question instead. Okay? Let's do that.
Dr. Hornstein, can you post this like slideshow onto Jupyter? Because
what I'm going to do, I'm not going to do that. What I'm doing is I'm posting the recording of the class.
Oh, okay.
So you have the slideshow. Okay.
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 Yeah.
Okay. Good. So I will see you tomorrow. We'll continue the lecture. We'll talk about Azaria Chamberlain. Any other questions? Okay. I'll see you tomorrow. Take care.
Dr. Hornstein.
Bye, Dr. Hornstein.
Bye.
Bye.
All ready? Okay, here we go.